Hello, everybody. My name is Anatoly Korkin, and today we have our 16 lessons from the series Atomic Scale Design of Molecules and Materials for Newbies. Today, I want to show you the new, very interesting instrument for illustration of laws, physics, chemistry, and biology at atomic scales. It's a very simple interface. It's based on web browser, so you don't need to download anything. And the illustration are pretty simple, so this lesson can be used at a lower grade of K-12 education. So let me just share a screen and show you the web interface. So it's called Next Generation Molecular Workbench. So, and if we, there is menu here, interactive. So that's basically the some examples of various laws, activities that where the students and teachers can have some tests to be done. And then actually there's important part which call more. There is classic molecular workbench where you can download quite a few interesting examples of laws, physics, chemistry, and biology. So let's go back to next generation workbench. Okay, so let's go to interactives. So if we click here and browse interactive, so we come to the section of showing physics, chemistry, and biology. And what is interesting and important to illustrate that there is similar sections on both area, which is called diffusion. So which is a good illustration that the same processes are studied both as physical, chemical, and also in biology with a certain specifics. Usually in the case of physics, it's atoms or basically the particles. In chemistry, it's part of the chemical reactions. In biology, it's also the process of some biological reaction, including the diffusions from biological membranes, where the molecules go from to inside of the cells or out of the cells. Which show the metabolism process. So let's just start from physics section, and I will not go into show you our all examples. So they're pretty simple. There is nice explanation. So that's basically the good to explore it by yourself or together with others, with the teachers and parents, whoever is interested to see it. So let's just click on diffusion. Okay, so again, there is a different subsection, and I'll click on diffusion and temperature to illustrate how diffusion is influenced on a temperature. By the way, I apologize in advance if in some cases screen may get frozen. It's happened sometimes when a few programs are working together. So, all right. So in order to start the illustration, I need to click on start, and you see there is a temperature here. Let's put it on low first. So let's show thermometer. So, and I'll start. So you see the green molecules or green atoms moving on one side and the blue on another side. So in this case, once I remove the barrier, so the blue molecules start penetrate in the right side and the green molecule on the left side, and they are mixed. And that's what they call the process of diffusion. Once there is enough equal molecules on all parts, the diffusion is becoming the equilibrium. So in order to accelerate it, we increase the temperature. So molecules start to move faster. So that's a good illustration of what the process diffusion is. So now let us move to the chemistry section. So there is different chemical reactions here. I would show you only one example, which is called catalysis. Catalytic reactions are the reactions are facilitated by a molecule, if it's in gas phase or liquid, or surface or clusters. Again, it could be clusters, could be in liquids or in a solid state. What catalyst does, it's either proceed as an intermediate by forming or clusters with molecule, and it's help to break some bonds. And then eventually catalysis gets separated from the molecules, but it facilitates the chemical reaction. So let's start the process. If we see nothing is happening if there is no catalysis. So here we click on add catalyst. So and once catalyst appear, it's break one of the molecules, and the reactive part is becoming proactive and can form the new chemical bonds. And once these bonds appear, so we see some energy release, and the molecules start move faster, and the process is going more actively, and you see more and more new molecule form. We can actually add more catalysts here, but you already see that most of the molecules from being green or blue, they started being mixed, green and blue. That's what illustrated in this process, and it's called catalysis. All right. So also in the chemistry section, I want to show you another important process, which is called phase change. Phase change is basically when over the temperature or pressure, the substance is changing from solid state to liquid states, like for example with high temperature, and becoming the gaseous at a high point. So let's just click on what called boiling point illustration. So here on the left side, we see the non-polar molecule, and on the right side, polar molecule. So if we start the process at a low temperature, so there's only a vibration process occur. So in this case, the right side is the solid state, and also the on the left side is also it's kind of more or less solid. It's also called like a gel part. But if we increase the temperature, adding the heat, so we see that molecules start break, and eventually they fly away from each other and form the gaseous state. They become gases. So and if you see that the polar molecules, they kind of more resistant to the process, and so they are evaporated at a high temperature. So this is illustration of the phase transition. So now let's go to the last part, biology, where it is. It's here, and here also will show you another example of diffusion, which is somewhat typical for biological process, where you have some molecules of different size, like, for example, inside or outside of the cell, and there is membrane with the pores. So if we start the process, you see there is pore size, which is large enough for the blue small molecules, but small enough for the green molecules. So if we started the process which allowed them to move, so we'll see that periodically the blue molecules penetrate on the left side. For example, that could be our transferring some molecules inside of the pore. So however, there is an instrument here, we can change the pore size, increase it, and now we can see the bigger molecules penetrate on the right side. And this is this size of the pore can be regulated by special proteins, for example, which can open or close the process of moving molecules in and out, which can illustrate, for example, metabolism of the cells. As a last example, I want to show you a very important process of formation of protein based on which are coded by DNA. And this process has two steps. The first step is called transcription, and we'll start it. And the second is translation. The transcription is a process which include two steps. First of all, it's break their double chain DNA on two processes with a protein called DNA polymerase. And then it's also forming their the messenger RNA, which participate in the next processes of protein formation. So here we see like RNA is being formed on DNA. So now if we click on translate, so we separate the new form RNA, ribonucleic RNA, and then with a so-called transport RNA, which bring the amino acids toward their RNA fragment and form the protein. So this is the way how RNA code the protein. So we'll see there are step by step the protein is formed. Now it's happily shaking. So that's an illustration of RNA formation. So you can browse all of this section, biology, chemistry, and physics, and see how the laws are illustrated by on atomic scale level. It's pretty very useful instrument. I like it very much. So that's about it for today. Thank you very much for your attention. I hope you like this instrument and use it in your lessons. Thank you so much. Bye-bye.